Good afternoon, everybody. Thank you for connecting to this new seminar from the LWNVIVAT series. It's my pleasure to introduce the speaker that we have for today. Is Dr. Jordi Figuerola. Dr. Figuerola is research professor at the Department of Biología de la Conservación y Cambio Global at CSIC en la Estación Biológica de Doñana. His research focuses on the interplay between behavior, evolutionary, and population ecology, and in the last year he has been investigating the transmission dynamic of different pathogens with a special focus on West Nile virus. So today, Dr. Figuerola is going to tell us about the West Nile virus surveillance in mosquito, provide useful information to reduce its impact and understand its ecology. So, Jordi, thank you very much for accepting participating in this seminar series, and the floor is yours.
Thanks, Jorge, for the invitation. And I will talk about some of the research we have done in the last years regarding the ecology of West Nile virus transmission. If I'm able to share the presentation. Okay. Okay, so all you know West Nile virus, you know that it's the Flavivirus more with a larger distribution in the world. It's present in all the continents and is a big source of public health problems. In the case of Europe, here you have a summary of the areas in 2024 where human cases have been recorded. West Nile virus is living and observed in the last years in Europe. Since the start of the 21st century, the incidence has increased, the geographic spread has increased, and we can consider that West Nile virus is now endemic in many places of Europe, like Spain, Italy, and Greece, where the virus is clearly overwintering and producing every year a high number of cases. This is the biological cycle of the virus. So birds are the primary host, the competent host. Many Culex species of mosquitoes are competent vectors, and the cycle is from birds to mosquitoes, and the problems happen when humans or horses are infected. They are not competent hosts. They are then epidemiologically, they cannot transmit the virus to new mosquitoes, but they can suffer disease. We have been working with West Nile virus for a long time, near 20 years. First, because the Spanish Ministry of Health was interested in the risk that West Nile virus was introduced by migratory species in Spain. And the first result of the project we started in 2003 was saying that you don't need to worry about the risk of introduction because the virus is already here, it's already in Spain, and you can see in the image the proportion, the prevalence of West Nile virus antibodies in common coots at the bottom of the slide, and in horses that live in Doñana, and you can see how every year we find prevalence of West Nile virus antibodies in these animals. And this annual variation is not random, but we have seen that it's associated with different climatological variables. So in the case of the horses, minimum winter temperatures are clearly associated with the prevalence of West Nile virus the next spring and the next summer. So after winters with more mild temperatures, we will find a higher incidence of West Nile virus. In the case of rainfall, the relationship is a little bit counterintuitive because in the driest years, we found a larger incidence of West Nile virus antibodies. And we think that this happens because the horses, the mosquitoes, and the birds concentrate in the few places where you find water in these natural areas where the precipitation is low. When we look at the common coots, we found a similar result with in the case of temperature. So when winter temperatures are higher, the next spring and the next autumn, we have higher prevalence of West Nile virus antibodies. In this case, we don't find any association with precipitation, and probably because they are birds, so they can move where they have water. So do not have the same problems that the horses that are closeted in a 3,000 or 5,000 protected area, and at the end they must look for the few places where they have water. These common coots can just change to another region of Spain with more water. Here you can see a little bit the history of West Nile virus. So in 2003, it happens the first detection in birds. In 2004, there was the first detection in human, a clinical case. From 2005, we are following the serology in horses, and we are finding seroconversions that support the local circulation. In 2006, 2008, we started to detect the virus in the mosquitoes. And what was important is that at the start, we were looking for the virus mainly in Culex pipiens because it was supposed to be the main vector in Europe. But until we don't look into Culex perexius, another species with a very similar ecology, we did not find the virus. And the question is that in Southern Europe, in southwestern Spain, this Culex perexius species is playing a big role in the transmission of the virus. In 2010, we had 2 clinical cases. 6 years later, 3 human clinical cases. So you can see, like as we had a lot of circulation in the birds, we were detecting a lot of antibodies in the common coots, but very few human cases. But in 2020, it changed. We had a first outbreak with 77 cases, 8 deaths. Then we had some years of low circulation, but suddenly last year in 2024, we had an increase of 158 cases and 20 deaths. The situation in 2025 is more controlled, and in the case of Andalusia, there have been a number of mosquito control operation and virus surveillance that probably is helping to control the West Nile virus circulation. And you can see how the number of cases have gone down from 118 in 2024 to only 2 cases in 2025. While in Extremadura, that's the region just in the north of Andalusia, the virus is circulating with a similar intensity to last year. All our work with West Nile virus surveillance in mosquitoes started in 2020, just because we were working with mosquitoes and West Nile virus, but not for surveillance. We were trapping mosquitoes every year, identifying to species level, and then in winter we were looking for Sutu and for West Nile to know how was the circulation, which species were more important. But as I said, in 2020, a big outbreak started. You can see in blue colors the date of initiation of symptoms of human cases in the province of Sevilla. In red, brown, the cases in the province of Cádiz, and in green, in Extremadura. And you can see how the outbreak is not detected until the start of August. Is this first black arrow is when it is detected that something strange was happening because 5 cases of meningoencephalitis were occurring in small villages, in village with less than 40,000 inhabitants. And 1 week later, the laboratory confirms that it was West Nile virus, and then the mosquito control operation starts. And the first important lesson was that the local companies of pest control were not prepared to make control of mosquitoes in urban areas. And this was a big problem. So in theory, by 15 August, the vector control operations were started, but the effectiveness of these first treatments was very low. So one important question is that at the same time, we were collecting mosquitoes, but we were not able to analyze the mosquitoes because of COVID. So all our equipment was in a hospital in Sevilla. So when we had the mosquitoes, but we didn't have the results. So when the outbreak started, we sent the mosquitoes to the National Center of Microbiology, and they analyzed the mosquitoes, and they confirmed that by 16 July, we had already trapped a pool of mosquitoes with the virus. Okay, so what is important here is that when the outbreak was detected by mid-August, most of the infections in Sevilla had already occurred. So this suggests that we need really good surveillance programs that are able to signal when and where intense circulation of West Nile virus is occurring. Because if we wait for the first human cases to occur, when we will be able to start to make control operations, probably the outbreak is fading out by itself. So this first detection in 16 July makes us to propose a preliminary pilot project to test the effectiveness of these mosquito surveillance operations. Okay, you can see here a little bit the calendar of what happened. In 13 August, the first human case is declared, but you know that usually West Nile virus surveillance is based on horse surveillance, and you can see here how the first horse case was not declared until 10th of August. So this suggests that really the surveillance of mosquitoes can be more effective than horses and can be more effective than surveillance in humans. As I said, our work with West Nile virus in mosquitoes, the first surprise was that Culex pipiens was not the main vector of West Nile virus. Most of the positive pools were detected in Culex perexivus. The infection rates were much higher in Culex perexivus than in Culex pipiens. This doesn't mean that Culex pipiens is not important, but what we think is happening is that we have a cycle in the natural areas that is highly dependent of Culex perexivus in these areas, and when the infected birds or even the infected mosquitoes moves into the urban areas is when the virus starts to circulate in Culex pipiens, and probably Culex pipiens and to a lesser degree Culex perexivus is transmitting the virus to these human cases. When we look at what species were involved in the circulation of the virus inside the villages, we trapped birds in the public parks, and what we found was that common blackbird, Turdus merula, was presented a very high incidence of West Nile virus antibodies. 91% of the birds was positive by seroneutralization. So this suggests that it's an important species, is a species that is highly preferred to feed by Culex pipiens. And you can see how other species are less involved, like the Sturnus unicolor and doves, the Streptopelia decaucto, and you can see how house sparrows have lower prevalence of antibodies. As I said, we started this pilot project. where this is the area where most of the cases occur, is Coria del Rio and Puebla del Rio. This is very close to Sevilla. It's less than 15 kilometers from Sevilla. And you can see all these areas here are 36,000 hectares of rice fields that at that time didn't receive any treatment against mosquitoes. And we think that this has been the main driver of these West Nile virus outbreaks in 2020 and 2024. So in each of these red points, we started to monitor 3 BG traps for mosquitoes every week from March to November. And in this web page, you can see in real time the number of species of females that are trapped in this area. We group the data by species. In pink color, you have the species that are highly competent for West Nile virus, that are probably more involved in the transmission of West Nile virus. And in blue color, you can see the species that are probably less important for West Nile virus transmission. This is how it works. We put the traps. This is the old model of BG traps. So who work with BG traps is now used to a blue trap. This is the previous version that is very effective, has been working by more than 15 years. So we trap the birds. We attract the birds with 1 kilogram of dry ice, the birds, the mosquitoes. This is a small capture of mosquitoes after 24 hours. We do all the process keeping the mosquitoes under cold conditions. We identify to species level. We do pools of up to 50 unfed females. We focus in 4 species that by previous studies we know that are involved in West Nile virus transmission: Culex perexivus, Culex pipiens, and Culex modestus. And we added Culex laticinthus that has a similar ecology, and we suspected that can be also important, and our results suggest that this is the case. So we do detection of West Nile virus by real-time PCR, and all the process used to be completed in 3 to 7 working days. You know that by chance, every time you have problems with a sample, by Murphy's law, it's most possible that this sample is positive. When we obtain a positive, we send the sample to the National Center of Microbiology, so we obtain the full genome of at least one positive pool by locality and by season. Here you can see the results of samples at the end of August in 2021, when we detected a really very high circulation of West Nile virus again in the area. This is a summary of the results of 2024. We analyzed near 200,000 mosquito females. We analyzed by molecular methods, so we identified by taxonomic methods many more. We analyzed near 7,000 pools, and as I said, 2024 was the year of a large outbreak, and we analyzed 3,000 pools of Culex perexivus, and more than 400 were positive. So 12% of the Culex perexivus pools we analyzed were positives. The number of Culex pipiens analyzed was lower, and the positive rates by pool was 1.7. Culex modestus is very important for West Nile virus circulation in areas like Camargue in southern France, but in our study area it's not very common. In Andalusia it's not very common. It's more common in Valencia and other areas of Spain. And in this case, we didn't find any positive species. And as I said, Culex laticinthus have a positive rate in pools similar to Culex pipiens, of 1.9. So this is how the surveillance work. So at EBD, at the Estación Biológica de Doñana, we trap the mosquitoes, we identify to species level, we do the extraction of the nucleic acids, and we do the real-time PCR. When we get a positive results, we send the sample for confirmation to the National Center of Microbiology, that is the reference center for West Nile virus, and we also communicate to the regional Ministry of Health. And all this is coordinated with the Spanish Ministry of Health, and the regional Ministry of Health is the responsible to communicate the results to the local governments and to the health delegations, so the hospitals and are aware that West Nile virus is circulating in the area, and the local governments can reinforce the message to the population to protect from mosquitoes and the control operations to reduce the abundance of the mosquitoes. The system works very well. Here you can see a summary of how the system has worked in the last years, and you can see how every year we are detecting the virus 1 month before the first human cases, in some cases even 2 months before the first human cases. This has been also the case in 2025. So we are able to not only say when the intensity of circulation starts to increase, but also to identify where it's most possible the first human cases occur. This is an example of how the amplification, the typical amplification in the areas near the rice fields area occur. And what you can see here is the number of females trapped by night in these traps. And you can see the black lines, the upper black lines are the number of females trapped in the areas in the rice fields. And you can see how the rice fields get water at the end of May, start of June, and then in July the numbers start quickly to increase. Note that the scale is logarithmic. We can reach near 10,000 females in one night. That's really a huge density of mosquitoes, of Culex perexiguus. And you can see how in the nearby village, 1 or 2 weeks before, after the numbers start to increase in the rice field area, the numbers start also to increase because the mosquitoes probably are moving to this village from the rice fields, and this green area corresponds to two other villages that are more far away from the rice fields. So there is a delay between the proliferation of the mosquitoes in the rice fields and the start of the increase in the urban areas. The control operations, mosquito control operations, were able to control the mosquitoes in the green line areas because they are far away from the rice fields. But in the case of Puebla del Rio, that is the traps with the red line, the village is close to the rice fields. Close means that there is one street where in one side you have houses, on the other side you have the rice fields. And in this area, the control was not possible until the treatment started to be done in the rice fields. Here in the bottom graph, you can see how the intensity of infection of the mosquitoes changed across the season. And you can see in this case how at the end of August, the intensity of infection in the village arrived to 1 of every 30 Culex perexiguus was infected by the virus, by West Nile virus. So the intensity of infection was really, really, really high. With the help of Fundación La Caixa, we started a new project to try to predict where and when we will have the larger proliferations of West Nile virus and of mosquitoes. So with the help of this Fundación La Caixa and different other administrations, this is all the areas we sampled in 2024. And the red dots and the yellow dots and the purple dots are sampled every week, and the blue dots and the gray dots are only sampled in 3 occasions per year. And this gives us a very good coverage of all southwestern Andalusia. That was where originally these outbreaks were happening, and allowed us to monitor the movement of West Nile virus. You can see the green dots are some traps that have been monitored in this week and were negative. And you can see how in 2024 the first positive starts to appear in June, close to the rice field area, and then other positive starts to appear in areas far away. Okay, and how the circulation by the end of October just disappears. Not because the virus disappears from South Spain, the virus is endemic, but just the intensity of circulation is not enough high for us to detect given the number of mosquitoes we trap. This effort to characterize the mosquito population is allowing us to develop Bayesian models on when and where we can find the larger abundance of mosquitoes. Here you have the prediction models for 2023 and 2024. 2023 was a very dry year that was characterized because the cultivation of rice was very restricted. Only 2,000 hectares were cultivated, while in 2024 we cultivated more than 24,000 hectares concentrated in this area, in close to Sevilla, where the main outbreaks are happening, and also in this area in Cádiz, where West Nile virus is always, is every year used to produce problems. You can see how different are the scales. So where in 2023 we collected 400 mosquitoes by maximum. In 2024, the maximum collection can be of 4,000 mosquitoes per trap night. And you can see how for Culex perexius, the distance to the rice fields is very important. But another important factor is the spatial autocorrelation. So Culex pipiens seems to have a lower spatial correlation, while the spatial correlation for Culex perexius is much higher and can reach near 40 kilometers, probably because the mosquitoes, day by day, just spread from these mosquito main breeding areas in the rice fields into the nearby areas and the nearby villages behind these areas and where the cases of infections to humans happen. You can see how temperature is important. Again, when we have less cold, we have more mosquitoes. And you can also see how precipitation is not important. And this is because these mosquitoes depend on the rice fields and depends on the irrigation lands. And the surface dedicated to these activities does not depend on the rainfall, depends on the quantity of water that it's in the embalses, that it's stored up the river. So if we have enough stored water to keep the cultivation by 2 or 3 years, even if we have drought time, we will have problems with the mosquitoes because of these areas. Another important thing we obtain from this surveillance with the mosquitoes is that it's much more easy to obtain the full genome of the mosquitoes, just because the viremia in humans is short duration and is lower, and in the mosquitoes, the viremias are high. So it's very easy to obtain, or it's much more easy to obtain these full genomes. And if we look at the history of West Nile virus in Spain in the last decades, we can say that all the cases we have observed in mosquitoes and in birds came from 5 or 6 independent introductions. And if we look at the history of these 2020 and 2024 outbreaks, the origin of the outbreaks were not new arrivals of West Nile virus, were West Nile variants that have been circulating in the area for many years before the outbreak. For example, the outbreak of 2020 was produced by a variant that has been detected in the area since 2016. While the 2024 outbreak was produced by another variant that also has been circulating in Spain from years before the outbreak happened. I don't know how we go of time. As I said before, I think we have developed a West Nile virus risk map for the government, so we can infer the risk of West Nile virus transmission at each different municipality. And for this, we have used the cases of human infection, of West Nile virus infections in humans, horses, and birds. And we have used layers of climate, land use, avian richness, breeding birds, number of species of breeding birds in each place, and vector distribution. For that, we first developed these distribution maps for Spain and also for all Europe, for these 4 main species that are involved in West Nile virus transmission. Culex inevitatus is a complex of species that includes Culex perexivus, and you can see how the distribution is restricted to the main affected areas. This is not true. Culex univitatus is not present in the Balearic Island, okay, but the Balearic Islands have the environment, the kind of environment that should allow the species to establish if it is introduced. But just because it's an island, the mosquito has not been able to establish. This is some of the layers we used. As I said, we developed these models for all Europe. And basically, what we found in Spain was that this is the risk of West Nile virus cases for all Spain. The redder the color, the higher the risk. And you can see how summer temperature, the presence of vectors, when more species of vectors are present, you have a higher risk of having West Nile virus cases. As we said, the temperature in winter is very important. When the winter is warmer, you will have a higher risk of West Nile virus cases. The runoff, that's the quantity of water that arrives to the place, how the habitat is transformed, and the bird richness are all related. You can see here the shape of these relationships. For example, in the case of bird richness, the factor is not the more important one, but you can see how the relationship is positive. And I remember that because there are many studies that suggest that the biodiversity reduces the risk of transmission, but all the studies we are doing in southwest Spain support the idea that where you have more species of birds, you have a higher risk of West Nile virus transmission. You can see here how what I said, that when summer temperatures goes up, the risk of West Nile virus increases. When the winter is warmer, the risk of West Nile virus cases is higher, and when the human footprint is higher, there is a larger risk, probably because it's at these more transformed areas where humans are living. And you can see here how the effect of runoff or the access to water is very limited. So if you are in a very dry area, you will not have a risk of West Nile virus. But as you increase the availability of water, the risk of West Nile virus increase a lot, but then remains stable. Okay, so water is important, but not in a linear way. And as I said, where you have more vectors of West Nile virus, you have a high risk of cases. You can check this model in the web. It's in the web of the Ministry of Health. You can access this web by the QR. You can also access all the protocol we use for this entomological surveillance. We have made a detailed description of all the methods we use for the trapping of the mosquitoes, for identifying the places, for identifying the mosquitoes, for doing the PCRs, all the protocols you can find here in this QR. The protocols are in Spanish, but thanks to Google and all these artificial intelligence methods, you can quickly translate to the language you want, and you can find information useful to you if you want to do this sort of surveillance. It's a lot of work, it's a lot of effort, but the results I think that are very, very positive. One question, if this surveillance is so good, why you have this large outbreak in 2024? And the reason is that surveillance is not enough. It's important that the control operations start as soon as the surveillance identifies the circulation. And that means that the administration must have decided before which task they will do in the case you detect a positive. And this was not working very well in 2024. And now this situation has changed a lot. And in 2025, I think that the system is working very well. You can see here all the places where we have detected West Nile virus circulation in mosquitoes. As soon as the virus is detected, the control operations are reinforced. The local administration communicate to the population the risk and which measures they must take to protect themselves from mosquitoes and to prevent the proliferation of mosquitoes in the area. And in this area last year, we had more than 118 cases of human cases of West Nile virus, and this year we have only 2 cases. So probably other factors are also important, like that many of the birds had already antibodies, and this might have reduced the circulation of the virus. But we know that, for example, here is Extremadura is having a similar number of cases in humans, is having even more deaths than last year, and probably all this control operation is not being done with the intensity that is being done in Andalusia. So to finalize, we can say that entomological surveillance allows early detection of virus circulation, that West Nile virus is endemic in Spain, so we need to control and to adapt to the presence of this virus. Blackbirds and house sparrows were involved in West Nile virus circulation in urban environments. This doesn't mean that we must kill these birds. It means that they are involved in the amplification and are good candidates for surveillance. West Nile virus circulation will continue in Spain during the next years. So it's not a question of bad luck. We have had bad luck in 2024. And if we keep doing nothing, they will solve by themselves. No, it's a problem that has arrived and will remain here. And the incidence in animals will increase. That's what we saw from the works with common coots and the horses. But the impact in humans will depend on how we manage this problem. And we have examples where the management of mosquitoes is successfully reducing the incidence of West Nile virus in humans. So it's important to prevent the mosquitoes breeding inside the village, but also it's important to control what is done in the peri-urban areas, because if we have activities that involve keeping flooded surface, we will have mosquito breeding areas near these human inhabited areas. So to prevent West Nile virus in the municipalities close to rice farming areas, it is important to implement mosquito control plans in these agricultural areas, or we will have problems in the nearby village. And that's what I have prepared for today's talk. So if you have any question.
Thank you, Jordi. Very interesting presentation and very interesting data that you share with us. We have time for questions, so if somebody in the audience want to address some question, maybe try to use the micro. I don't know if today the micro is working. No? If not, I can start making some questions to Jordi. Jordi, I have a question regarding to the species of the mosquito that you comment. Do you know if depending on the species of the mosquito that is transmitting the virus, there is any particularity of this virus regarding to the pathogenia that can cause in horse or in human?
No, we don't have data showing that particular variants of the virus are associated to different species. Probably all the species are in one way or the other involved. So where you are in the more farm areas or rice farming areas, you have a high role of Culex perexiguus. But in the village, these mosquitoes mix with Culex pipiens, and there are lots of other places where Culex pipiens is more abundant because Culex pipiens prefers more freshwater areas than Culex perexiguus. But I don't think that we have virus variants associated to these different mosquito species. In any case, we are producing the genomes, so we hope we can check these sort of things in the near future.
Thank you, Jordi. We have some question in the chat. Heidi from Italy is interested to know about the correlation that you observe between bird species diversity richness and the West Nile virus prevalence. She said, might it be that you have a lot of migratory birds, especially in Doñana, whereas other study habitat with more stationary habitats?
This results I have shown is from all Spain. It's not restricted to Doñana. We made a previous study where we analyzed 45 localities in southwestern Spain. Some of the places were in Doñana, but many others were outside Doñana. And in that case, we also found this positive relationship. And when we look at the breeding diversity, yeah, it's true. It's larger in other places. We need to check. In general, I will say that while most of the support for this dilution effect came from North America, the studies done in Europe are not so clearly supporting this dilution effect.
Okay, thank you. Carlos, can you try to use the micro and ask directly?
Yes, I think now it's activated. Do you hear me?
Yes,
Yeah.
we can hear you.
Okay. First of all, Jordi, congratulations for your excellent job, because it's really important to have in Spain such this group working in the problem that we have with West Nile virus transmission. And I have many questions, but I will ask you about two. The easiest one is, how do you identify females of Laticynthus from the rest of the Culex species?
By morphology. I know it's difficult, but we use morphology to separate the species.
I think it's almost impossible, but okay,
yeah,
so you have to teach me how you do that.
It's difficult, and we check by genome, by COI, and we don't fail so much.
Okay, and the other is about what is happening now in 2025.
Yeah.
You say that in this area, in Coria del Rio and around, you have more than 36,000 hectares of rice field pads.
Yeah.
So how many hectares, do you know how many hectares have been treated in 2025?
Yeah, there have, the initial plans were 6,000. That were the initial plans, but the reality must have increased this surface. I think it has been increased to 8,000. And even with 8,000, treatment with BTI, with Bacillus thuringiensis, with larvicides. That's not enough. At the end, you need to do adulticides to protect the village, because the production of mosquitoes is very high.
So, and did you find any reductions on your catches of mosquitoes? Because last year you said almost 200,000 females, and now do you see a reduction on the number of females?
Yeah, we see the reduction. As I said, not as general as we would like. So we see the reduction in the village, and in these general places, in these Cañada and Dehesa places, the reductions were not so clear. And but since the numbers were increasing a lot, then they started adulticide programs, not focused on the trap sites, but in the general area, and there were significant reductions. But I must say that if the treatments are not repeated every 2 weeks, you have increases.
Yeah, okay. And the very last question, you perfectly say that there is a difference between surveillance and control. Surveillance in Andalusia is wonderful because you find the virus in mosquitoes 1 or 2 months before there are the first cases. This is something very, very useful, very interesting. But do you really think that the reason why in Andalusia now you have so few cases is only or mainly because the reduction of the mosquito population, or do you think that there may be other factors? In other words, do you think that in next year, if there is a good reduction of mosquitoes, also there will be a reduction on cases of West Nile virus?
We have had something that has helped us a lot, I suspect, that is the circulation of West Nile virus last year was so intense that we detected seroprevalence of West Nile virus antibodies of 100% in the blackbirds and up to 60% in the house sparrows. So my feeling is that what we are finding now this year is a combination of 2 factors. So these larger control operations that is contributing to reduce the transmission, but also the fact that until the juveniles have not fledged yet, the susceptible birds, the proportion of susceptible birds to West Nile virus was lower because of the high circulation last year. I think that both things are contributing to that when we look at what is happening in Extremadura, because Extremadura also had a very intense circulation last year, and this year the number of cases is a little bit lower, the number of deaths is high, and the circulation in the mosquitoes is very intensive. So what we were finding last year, this proportion of positives in the mosquitoes that we find in 2024 is being found now by the people that is working in Extremadura. That last year were not working in the area. Extremadura has started a similar surveillance project, not so intensive, and they are this year finding, even after the strong circulation of last year, they are finding a lot of circulation in the mosquitoes. And this is the reason why I think that, okay, the seropositivity in birds is helping, but also the efforts to control the mosquitoes are also important to control that.
Thank you, Jordi, and congratulations again.
Thank you.
We have other questions from the audience. Have you considered to investigate the blood feeding pattern of the mosquito?
Yeah, Yeah, we have doing that. So I'm very happy that you. I just deleted. You can see the slide.
Yes.
Okay, this is what the cool excipients fit inside Puebla del Rio. That's one of the villages that has been very affected by the outbreak in 2020 and 2024. And what you can see here is that the proportion of cool excipients blood meals on humans is very low. If you compare this with other places, used to be 5%, even higher. In Barcelona, for example, the proportion of blood meals in humans is even much higher. This is what I explained before, that the high preference of cool excipients for blackbirds, 70% of the 70.6 of the blood meals came from blackbirds, also house sparrows and greenfins. Here you have the chickens that are not competent. This is why they are on this side. And here you can see the large diversity of mammals that are important. One important thing is how important pet species are. So 10% on cats, 11% on dogs. You must think that we are in a very rural area where many people have horses at their homes or at their houses. There are rats, bats, fox, donkeys, and cows. So the origin why we decided to study this species of mosquitoes and not others was based on a original study on the blood meal feeding preferences of the mosquitoes that allowed us to identify the species that were feeding a lot on birds and that also have high vectorial capacities.
Okay, thank you. Another question is, how do you isolate the virus to investigate if there is any mutation that could be related to increased pathogenesis?
We have all the genomes, and the Centro Nacional de Microbiología is isolating the virus, but we have not done studies of pathogenicity yet.
Okay, thank you. And would it be possible to combine genotyping with molecular clock?
Yeah, we have done that for some years ago. We were trying to get the genome of one pool of mosquitoes, and then we discovered that the sequences we found were part of West Nile, but there were a lot of Simbis sequences in this pool. So then we realized that 12% of the pools in 2022 in Andalusia were infected by Simbis that had not been described in Spain yet. And we used molecular clock to infer that the introduction of Simbis in Spain probably has occurred 5 years before, by 2017. So we are planning similar analysis for West Nile virus. And what interests us a lot is how the different virus we isolate at the different places in different years, how they are related.
Okay, thank you. We have another question that is related to the co-circulation of other orthoflavivirus, like dengue, slave, busk, on those areas where West Nile virus is prevalent, as it was initially hypothesized that the co-circulation of this related orthoflavivirus might confer overall protection. What do you know about the co-circulation of other flavivirus in this area?
We have Usutu. We are starting now a project to extend this analysis to Simbis and to Usutu. Simbis will not give cross reaction, so probably it's Usutu the important. Dengue is not circulating, or we have some immigrant population that has antibodies against dengue, but the prevalence in the population is really very low. and at the end, humans are not important for the amplification. So at the end, what it's important is not the antibody in the human population, but the antibody in the bird population. So probably the more important one will be Usutu, or could be Usutu, that we know that is circulating, and we know that even in some years, the intensity of circulation has been 10 times more higher for Usutu than for West Nile virus. But this was a lot of years ago. This was by 2010 or something like that. Usutu was circulating a lot.
Okay, thank you. Jordi, I have a question regarding to the serological surveillance that you are performing. How do you distinguish antibodies against Usutu and antibodies against West Nile virus?
Yeah, we are doing these studies in collaboration with Miguel Ángel Jiménez from INEA, and what we use is seroneutralization. So when we get a positive or doubtful by ELISA, we send the serum to him, and they make a seroneutralization in parallel against West Nile virus, Usutu, and Bagaza to see
Okay,
which.
so the confirmation is by neutralization.
Yeah, and it's important to seroneutralize, to make a confirmation by seroneutralization, because some years a significant proportion of the positives by ELISA could not be confirmed.
Okay. Okay, and the ELISA that you are running is a commercial one, or
yeah,
or
it's
a...
Ingenasa
Ingenasa.
ELISA.
Okay.
It works very well. So it's not, if you use the one from IDBET, you will have the same problem, that you need to confirm the ELISA positives. You need to confirm them by seroneutralization.
Jordi, I have another question. I have the curiosity how this virus is surviving to the winter. I mean,
Yeah.
how the when we are talking about the endemicity of this virus, we know that the season of the mosquito is when the transmission is occurring, but what happened with the reservoir of this virus in the nature during this period in which the mosquito is not so active or is inactive?
Yeah. There may be different mechanisms that may be at work. For example, it was not. I think it was not this winter. Last winter, there were detected 2 owls that died because of West Nile virus in January. So there is the potential for long-lasting infections, for example, in internal organs. So when raptors depredate, the infected animal can get infected. That's one possibility. In our area, mosquitoes are active all the year round. So we are finding females with recent blood meals in December, in January, in February, all year round. We have not been able to find the virus yet on these mosquitoes, but we have also estimated that if we want to be sure, so given the intensity of circulation of West Nile virus we detect in November, that's the last month we are detecting the virus, but given the intensity of circulation of the virus in November, we should test, I'm not sure of the number. I think we estimated it was 15,000 females. Is the number of mosquitoes we need to test in January to be sure that the virus is not there, with the intensity of infection of circulation we detect in November. And nobody has done this. You just are not able to trap 15,000 mosquitoes in January. The densities are very low, so probably they can survive in that way. There are publications in Czech Republic of infected mosquitoes wintering in cellars. So the mosquitoes were trapped underground and were infected with West Nile virus, and probably there are several mechanisms that may allow the mosquitoes or the virus to survive in the mosquitoes.
Okay, thank you. My last question, Jordi, is this is a virus that is transmitted by the mosquito and is circulating among the birds, but the virus can also infect on mammalian, not only human and horse. Do you think or this know if some other species of mammalian species can also work in the transmission of the virus, or all of them are considered end host?
In Western Europe, I will say that all of them are considered end host. There are some hamsters, I think, or animals similar to hamsters that are competent hosts, but they are not present in Western Europe. So in our area, should be birds. Later, then you have amphibians and reptiles. That is this lineage 5 that seems that is not pathogenic to humans and seems that can replicate a lot on amphibians. But to the moment, it's not a health problem in humans. But we don't have any idea of what can be the contribution of reptiles to lineage 1 or lineage 2 transmission. What we know is that we are detecting antibodies, for example, in lots of cows, for example, that apparently don't suffer West Nile virus infections, but we are finding lots of antibodies in the cows, for example.
Okay, thank you. So if there is no more question, I would like to thank Jordi for the excellent presentation and for the data that you share with us. It's very useful for us to know what is the situation in Spain and also in Europe. So thank you. We have another question for Nuria. Nuria, can you ask directly? Nuria, we cannot hear you.
Sorry, can you hear me now?
Yeah, yes, now yes.
So really nice presentation. So my question was related to the strength that you need to obtain in your neutralization assays to be really sure that you are having an activity. What is the range of dilutions that you need to surely confirm that you are having a neutralization capacity? What's your cutoff? That would be the question.
We are using a cutoff of 10.
Okay.
And to confirm, so we run the neutralization in parallel to SUTU and West Nile virus, and we require a difference, I think it's of 3 times to confirm that is one or the other.
Okay.
If not, it's
yeah, it can be tricky, right?
Unidentified Flavivirus. I think so. We need better tools for serology in humans. That's a problem, but also for birds, we need better tools, because at the end you have these problems that really you don't know. Okay, you have some samples that are really very clear. So, for example, we have retrapped birds that didn't have antibodies, and 1 month later they were neutralizing at 614 dilution. Wow. So okay. They have been exposed to the virus, but most of the positives are with 20,
okay.
And it's very hard.
Okay, no, thank you very much. That's very useful. Thank you so much, and congratulations by the excellent job you're doing.
Thanks.
Thank you, Nuria, and thank you, Jordi, for this job that you are doing. And if there is no more question, we can finish here this session. And again, thank you very much for joining this seminar. Thank you. Have a good day. Bye-bye.
Bye-bye.